Okay, I'm set. All right, everybody. I don't really see everybody. That's a pity, but anyway, there it is. I'm going to open the chat. Okay, so let's get started. Welcome, everybody. So for those of you who haven't been to any of my presentations yet this summer, my name is Carolyn Price, and I'm a faculty member in the Department of Cancer Biology. I also work in the Office of Graduate Education with Matt Schausel and Cindy Bukursky, who are here with you today. And basically the office is of graduate education is the one who is the office that's coordinating this virtual SURF. So I'm here to tell you about how to read a scientific journal article and also to talk about how to give an effective journal club presentation. So the talk's going to be split into two parts. And then later in July, those of you who are interested, you can join me, and depending on how many of you there are, some other faculty members in practicing giving a journal club presentation. But first of all, funny, my keys no longer control it, Matt.
How about a mouse click? Does that?
Yeah, it works. That's weird, is it? Zoom keeps changing stuff.
Yeah, I was going to say another update by Zoom. So.
Right, because the mouse used to, the keyboard used to work. Okay, sorry everybody, but I can't keep up with Zoom. They zoom along at a rapid speed of changes. So anyway, we have a chat box that I have open on my computer, and I would like your ideas about why you would read a scientific paper. So I'm going to give you a minute, and why don't you write down your thoughts about why you would read a scientific paper. There's some reasons that are really obvious, but there's other reasons that might be less obvious to you. To learn about a specific topic, to stay updated about findings in your field, to learn new results in your field. Sometimes mainstream media source misreport or misconstrue scientific findings, and it's important to go to the source to validate your findings or to give a more accurate picture. Yeah, how about reading about wearing masks? I went to a very interesting talk on that. It hasn't reached the literature yet. Yeah, investigate avenues for analysis. That's really important. New ideas to see if your hypothesis has already been researched. To find an experiment to reproduce or to learn about a new technique, or you want to write a review paper. Yeah, so you guys have got it. There's a lot of reasons to learn about the latest discoveries, to get educated about a topic, hear opinions of other scientists. Right, so there's a lot of different reasons. And just like there's lots of different reasons for reading a paper, is that there are also different types of papers that have really quite different slants. So let's take a look at this. So there's two types of papers that I kind of lump together as original research. And you can have full research or clinical reports, because you have clinical research and you have basic science research, translational research. And these are usually fairly long, detailed articles that report a whole series of experiments and the outcomes. Then you have shorter communications or brief communications. These are usually short research reports. It may be that the authors need to get some really exciting new results out rapidly because somebody else might publish first, but they tend to be shorter articles and somewhat less complete, but nonetheless important. And I have here peer-reviewed. A very important thing about these research articles is that they're peer-reviewed. So for those of you who may not have heard this term, what peer review means is that if you write a paper, your paper, before it's published, is sent out to other scientists who will read it and critique it and make suggestions for improvement. And we're going to talk a little bit more about the peer review process later on. But basically, it means these papers have been through some form of quality control. So getting back to the types of papers, another sort is a case report. This is usually a clinical paper, and it might be about a single case or a single patient, something unusual or interesting about that patient. Then there are techniques or methods papers. That might be somebody has developed a new technique in their lab that they've published in a research paper, but they might be invited to write a technique paper that gives more detail about how to get this method to work or other uses of it. For example, there were all sorts of CRISPR techniques papers when it first came out, and there still are as the technique evolves. Commentaries, they tend to be a short article with the author's opinions. Sometimes they're invited, sometimes the author will just write it and see if they can find a place to publish it. Editorials, they're usually invited, and they're often a short review or a critique of a recent research article. It's often a high-profile article that the editors of a journal want to highlight. And then there are review articles, and review articles are articles that summarize a whole body of work on a specific topic. So sometimes review articles are peer-reviewed. Some really good journals will send them out to be read by other experts in the field before they publish them. Other review articles are not peer-reviewed, and so you need to be aware of that the quality can vary quite a bit. And you'll see that as you read them. Review articles. Okay, so what's a paper made of? It's divided into sections, and most papers have a similar design. Not identical, but generally similar. Even though you have these different sorts of papers, they tend to be split into, there's a title. So here's an example. Can you see my mouse okay?
Yes.
Okay, good. I have a question here. Can you easily find out if a review article is reviewed or not? You know, that's not so easy, actually. No, it's not so easy. I'm sure you can find out, but it's not very easy on the surface of it to know whether it's being sent out for a review or not, in fact. So that's a good question. You'd have to look at the journal policies, but it's not necessarily written, whereas original research articles, they nearly always are peer-reviewed. So the organization of the paper, so you have a title, you have a section showing who all the authors were on the paper, then you have an abstract or a summary, which a little bit of it's shown here, and this summarizes what the paper is about. Then you tend to have an introduction or a background section, which basically gives you a review of the material that you need to understand to be able to understand the next sections, which are the results section and the methods or experimental design. Sometimes the results come before the methods and experimental design, sometimes they come after it. It just varies. And then you usually have a discussion section. And then, of course, references. Particularly for the introduction, the authors will be referring to other people's work, and that all needs to be referenced. Let's take a look at these sections. And you may have supplemental data, and we'll come back to that later, but this is basically can be very important experiments that don't necessarily fit into the main body of the paper. Let's look about look a bit more at these sections of the paper, because it's important to understand what the sections are about because it shows you where to find things in a paper. So the abstract, as I mentioned, is the sort of overview of the whole body of work, but it can be very dense and hard to read because it is a mixture of results, background and conclusions, all rolled into something that can be between 150 to 250 words. So it's pretty dense. So then you have the introduction. The introduction gives you the background information. It'll tell you, for example, the rationale for the study, particularly the last paragraph, and it will usually give you a very brief summary of the results. So the first paragraphs of the introduction basically give you the background information, the knowledge that you need to understand the paper, the key points. But then the rationale for the study and the summary of results, you go to the very end of the introduction and there'll be that last paragraph that just kind of, again, summarizes where this paper is going as a lead-in before you get into the results section. Then you have a methods or experimental design. This is kind of a standalone section which explains exactly how the experiments were done. The results section tells you the results of the experiments, and it explains enough of the experimental design so that you can understand what cells were used or what animals were used and who was compared to what, but it doesn't necessarily give you the nitty-gritty of exactly how many animals or how the cells were cultured or how they were counted, that sort of thing. That's where you go to the experimental design. So the results will be divided into sections, and each section usually corresponds to one or two figures that show the actual data. Then the discussion has the take-home message. There's a very important first paragraph of a discussion, which really summarizes the results and the take-home message of the paper. Then the discussion will get a bit more wordy usually. It will talk about why the study was important. It might relate it to past work. And then it often goes on and talks about what needs to be done next. And so if you are having to do, if you're entering graduate school, for example, and you have to come up with a project to defend for your PhD qualifying exam, the discussion section is a great place to look for future avenues of research. Supplemental data is really important. It often has more materials and methods. It may have a lot more results because journals now, they try to keep the papers fairly short, which there's a problem in that because it means there isn't room necessarily to explain all of the methods and experimental design in a lot of detail. It can't show, there isn't room in the results section to show all of the controls, for example, or some of the less important, not important, just some of the less key experiments. You might be limited to 4 figures in a paper. That's not unusual in some of the cell reports, for example. But to actually get those 4 key figures, you might need another 8 panels of figures to show that you've done everything, all the right controls, all the tangential experiments, and that all goes in the supplemental data. And if you really want to get to grips with a paper and how it works, you need to go through the supplemental data. So here's a kind of fun thing, the anatomy of a scientific paper. So you have your title, Are All Apples Red? By Ida Cortland, right? Here we have the abstract. We examined several apples' color. Although most are red, some are not. Here's the abstract. The introduction. An age-old question is, Are all apples red? And here we've got some references. Mr. McIntosh, 1993, thought so. G. Smith of 1999 disagrees. The method. We went to the local grocery store and bought one of every apple they had. We took them home and we looked at them. Here are the results. We found 4 red apples, 1 green apple, and 2 yellow apples. The discussion, summarizing the results, drawing the conclusion, since we found 1 yellow apple and 2 green apples, it must be true that all apples are not red. We concur with G. Smith's finding, and then here are the references. So it's a silly example, but I think it kind of illustrates the sections of a paper. Okay, how do I read a scientific paper? I've taken you through the different sections of the paper and the general order that they tend to appear in. I forgot to put materials and methods in here: title, abstract, introduction, results, discussion, supplemental data. I left out discussion as well. So do I just start at the title and read all the way through? Absolutely not. That is not the way I read a paper at all. It's far too much effort to read a paper that way and then discover that it wasn't the paper I wanted to read in the first place. So the way I go about reading a paper is, first I have to find out if I'm interested in it and if it's relevant to me. You know, it takes, as I'm going to talk about, it takes quite a long time to actually read a scientific paper properly, to digest all of the material and think about it. So first of all, I take a look at the title. That's the first guideline, what it's about. Then there's the abstract. But as I said, it can be a little dense because it's just got so much stuff interwoven into it. So if I find it hard to follow the abstract, I look at the first part of the introduction. That's a really good background. You know, if you're not so familiar with a subject, the very first paragraph of an introduction, that tends to give you the most basic background as a lead-in. Then, if I want to find out what the paper is really about, I will look at the last paragraph of the introduction and the first paragraph of the discussion. Remember, I was telling you that the last paragraph of the introduction usually sort of summarizes what the question that's being asked and the general approach to how it was asked. And then the discussion, the first paragraph of the discussion, usually summarizes the key take-home message without too many experimental results. And so if all of this checks out, and I think that this paper is relevant and is something I should be reading, then I have a very definite strategy for reading the paper, because, as I said, it takes a long time to actually digest everything. So what I'll do is I'll go through it very quickly the first time round. So I'll read all of the introduction or the background. You know, if it's something that's really in my area, my lab studied telomeres for years, and so I've read an awful lot of papers that start with the telomere is the natural end of a chromosome. It serves to protect the ends from blah, 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 right? I can still recite it. So I may not need all of the introduction, but I will read through it quickly to make sure I'm kind of in the right frame of mind, reading frame, thinking about the right things. Then I will read through the results and go through the figures. But again, I'll go through it quite quickly. It's kind of like taking a look at a map to find out where you are and seeing where you need to get to, but you aren't really plotting out your route in a whole lot of detail. Then I'll read the discussion and then come back to the abstract. So by now, I have a good idea, a good figure, a feel for what the paper's about, the types of experiments that were done, and what the authors are concluding. So, you know, I have a sort of a superficial feel for the paper, but I won't fully understand all of the experimental details, and I certainly couldn't give you my opinion of the work or use what I've learned to design experiments for myself, you know, so I haven't really properly absorbed it. So that takes me to stage 2, which is the slower stage. And this stage is where I will go back over the results and the figures one by one. And as I'm doing that, I will consult the supplemental data. So this is where I'm really thinking and analyzing the data, thinking about what the authors have said, seeing if I agree with it. And as I do this, I'll make notes as I go along. I'll make notes for each figure or section of a paper. A section may correspond to a couple of figures, or you might have one figure per section. It just depends. And I'll make a note of, you know, what was the experiment? What was the question? What was the experiment? What's the take-home message from the figure or this section? And then do I have any questions or concerns? So I will put a star, but I don't believe this, or but they didn't do so and so. So this is my notes, and I'll keep these notes. They're very useful to refer back to. Then I will reread the discussion and think about it in the context of what I've really learned for the figures. And then I may make some more notes about my thoughts and what, you know, what I've really learned from this paper, and what I think it means and where I think it should go next. So this takes time. I don't just go from start to finish, like I said. And it's worse than that. It's not just that I sort of jump around between sections, but I actually may have to take a lot of detours en route. So for example, if it's in a paper that's a bit outside my area, I may have no clue, right? And I expect a lot of you guys, you know, you're new to science research, and you're sort of dumped into a new area, and it's sort of, I don't even understand the words here. So what do? Well, I'll tell you a secret. I go to Wikipedia often. I don't necessarily believe Wikipedia, but Wikipedia is a great place to start. It's a sort of simple, basic explanation that gives me a jumping off point to delve a bit more deeply. So Wikipedia is definitely not the only source. It should never be the only source, but it's a good jumping off place. And then if I need a bit more background, I'll read a review, an article, like we talked about review articles, or I might consult another paper, or I might have to consult a whole lot of papers. This is why it all takes time and why I'm talking about detours. Then I might find this whole thing is really interesting, and I might want more detail, more perspective. So I might go and read other papers on a similar topic. I might go back to older papers on this topic. I might read a commentary. And so I can find that I've wasted half the day on this. Well, wasted isn't the right word, but it can be, you know, it can take quite a lot of time to really delve into the literature. And then eventually I'll be coming back to the paper that I started to read in the first place. So, as I said, getting to grips with the paper takes time, but you know, it's okay because it's really worth the effort. This is what gives me inspiration. You know, sometimes when I needed to think about my own research, I'm distracted, I've been busy with things, and I can't really think about where I need to go next. And I found over the years that the most stimulating thing to do is to sit down and read a new paper in my field. And that just kind of gets me thinking and gets the creative and inventive juices flowing. So, as I say, it takes time, but it's worth the effort. So anybody have any questions before I go on to the next section about giving a journal club? No questions? How many people here have been, I can't see everybody. How many people here have had to read science papers? You see that you've got this little reaction button. down at the bottom, if you hover at the bottom, how many of you have had a go at reading scientific papers? Put your hands up. Yep, quite a few of you. And how many of you have found it hard? I bet a lot of you. It's hard at first. And I've had first-year graduate students come to me. They had to read papers for their class, and they're saying, oh, it's so hard. It takes me so long. You know, what can I do to speed it up? And basically, it just does take a while, take a long time at first, but you get faster at it. And I think that if you put into practice some of the techniques that I've been telling you about, you'll find it easier, especially the splitting it up into the pieces and then writing down the conclusions for each piece. Okay. Back to this. Okay, so we're going to talk about giving an effective journal club. And so it's back in you guys' court. Why would you give a journal club? So tell me some reasons in the chat. So journal club is where one person presents a paper to the rest of the group. Why would you want to do that, do you think? Yeah. So what is a journal club exactly? To share what you've learned, to see how others interpret the same information, gain perspective on information, to get the benefit, to others can point out what they disagree with, to present scientific information to our peers. Yep, these are some of all of the reasons where the scientific literature is huge. And it's very difficult for everybody to keep abreast of all of the papers. And so it might be that you have a lab, my lab, sometimes we've had where everybody read the paper, but one person was in charge of presenting it and delving into the background so that they spent more time on it and could answer other people's questions. Other times we've wanted to study an area and everybody's presented a different paper, and it's a sort of quick way of bringing everybody up to speed. Before I go further into this, I wanted to see how many of you think you might be giving, joining me for a journal club presentation, practice journal club presentation in, was it 2 or 3 weeks? Could you put your hands up? Because I need to know how many people I need to. We've got 1, 2, 3, 4, 5, so far 6, 7, 8. All right.
There's a couple on the other. There's almost 2 screens worth of names. So just so you know, there's 4 on page 2.
Okay, 4, 5, 6, 7, 8. 8 people. Okay. I tell you what. How many papers we discuss and how many leaders we have depends on how many people are going to sign up. So I think what would the people who, at the end of my presentation today, the people who are interested in this, it might be good if you were just to email me directly. Matt will have the sign up and you can sign up there, but I think it would also be good for you to email me directly and tell me what area you're interested in. If there aren't very many people, we'll just have one group, which will probably talk about cancer biology. If there's more people than that, then we might have one on neurobiology, or we might have one on sort of developmental biology. It'll just depend on who else I can find to lead a group. But anyway, I think it would be good for you to sort of email me your interests. But let's get back to journal clubs. So let's talk a bit more about the main purpose of a journal club. And a lot of you have summarized these main points. It's a good way to bring members of a group up to date on an important advance or a new idea. It's also a good way to introduce a new technique or a clinical approach. I remember, this is really a long time ago, giving a journal club on PCR, when PCR was brand new to the world. And then in my lab, we had journal clubs on CRISPR, when that was brand new to the world. And this is how you learn about these techniques. And then you think, well, maybe I could use them in my research. We use journal clubs all the time for our graduate students. Our beginning graduate students have to participate in journal clubs because it's a good way to think critically about science and develop the ability to decide whether experiments are well done or whether controls are missing or what needs to be done next. Sometimes it's used, people can give an update on their research, or sometimes job candidates would have to give it. That's less common. Other purposes provide historical development of a field. This is quite useful. In my lab, sometimes we would go back and read old papers that had become really relevant due to recent publications, and we wanted to take another look at this old work to remember what had been shown then, and then we would read the more recent papers, and it kind of gave us a full perspective. Introduce somebody in the field, discuss applicable regulatory, ethical, or safety issues. Yeah, sometimes you'll actually read bad papers to look and see what the problems are. Some of you were probably at my ethics talk and some of the bad coronavirus papers. It'd be pretty interesting to have a journal club looking at some of those and picking apart what the problems were. And then, you know, we did this in my lab, and it was quite a good way for everybody to be talking together, interacting, talking about science, so to sort of build the group. So I've been part of a lot of different journal clubs. When I got my first faculty position, my lab was really small and I didn't have many people to talk to scientifically. I was in a chemistry department, but we had this great cross-department journal club in the evenings on nucleic acids that I went to for years. I went there for 12 years until, this was in Nebraska, till I moved to Cincinnati. It was great. Then one stage we had a summer journal club because there weren't any seminars going on. And we decided, well, let's make use of the summer to focus on new techniques. And so everybody, this was multiple labs participated, and everybody presented a new technique once a week. Well, one person a week. Then, as I've talked about, our lab meetings, where we sometimes had look at single papers, which everybody read, or we might have a theme where everybody's presenting a different paper. We talked about student data critiques. Also, I'll have students read papers in class, and each student will present a different figure and talk about, you know, what was the question that was being asked, what did the data show, and was it good, was it bad, do you have any critiques of the data? Okay, so how do you give a great journal club? It's intimidating, isn't it? You know, especially the first time around, you're not used to public speaking. There may be other people there who know more than you. Ooh, it's a bit scary, the big guys are there. But, you know, there's one good answer, and it's to prepare well. And if you prepare well, all will be well. So the very first thing is that you need to choose a great paper. You can't give a great presentation on a crummy paper. So here's this list of papers that we talked about, from the full research articles to the review articles, editorials, and commentaries. Okay, so which ones are good for journal club presentations? Well, let's think about what you're trying to get across. So first of all, the topic has to be relevant to your audience. So you're going to know who your audience is. And so it needs to be relevant and something that's going to interest them. So it's got to have new material or new ideas. You know, you don't want people to be sitting there that, well, who cares? You know, why is this person even subjecting me to this? So it's important to think about your audience. Then the other thing is high quality science. For a while, it seemed like people in my lab, they were picking these awful papers in low quality journals and really poor quality science. I'm not sure. I think it was because it was easier to present these short little papers rather than the more complicated, more important big papers. But I finally stopped allowing people to choose papers for a while until we got back on track and started talking about some high quality science, because it had been such a waste of time. So, which types of papers are good for journal club presentations? So I would say the full research reports and the clinical research reports or the brief communications tend to be very good, in part because they're peer reviewed, and so you have quality control. But if you're a clinician, then case reports might be really useful as well. You might have a patient who had something very unusual about them, and you could learn a lot from that. A techniques or a methods paper. As I said, we had one summer where we went over new methods papers. That can be really useful. Things that we don't normally use in a journal club are commentaries, editorials, or review articles. Now, you may go to review articles, editorials, and commentaries to gain the background and perspective for your presentation, but they would not normally be the focus of your presentation. I want to go back to peer review. What exactly is peer review and how does it work? And scientists love it and hate it. We love it because it generally makes for a much better quality science. We hate it because people are criticizing our research. We think our work is perfect and it's ready to publish, and other people tell us, oh, you need to go back and do this experiment and that experiment, and we don't want to do that. But in the end, generally it's good and it strengthens the science. So the way it works is a scientist studies something. Here they're studying stars or the moon or something. They write about their results and they send it to a journal. And you pick a journal that publishes in the area that you're writing about, and there's a certain amount of judgment which journal you think is suitable. You send it to the editor, and if you're lucky, the editor looks at it and thinks, oh, this looks good, this looks interesting, and sends it out to a bunch of your colleagues who will read it. And these are called peer reviewers, and they will read the article and provide feedback to the editor. The editor usually will then send the reviewer's comments back to you, and if the comments were reasonably favorable, the editor may say, okay, I'll publish it if you make these changes, or they may say, well, I'm going to reject it for now, but I'll reconsider it if you make these changes. Or if you really are out of luck, they may say, well, no, it's not suitable for this journal, or it's got problems and we just not going to publish it. In which case you try another journal, but though you might do a bit more work first to sort out the problems. Eventually, you have a lot of, even this can be several rounds of back and forth, and it can take a lot of months. It's pretty frustrating if it's you that's trying to publish the paper, especially, you know, if you're looking for a new position or trying to write a grant or something, trying to finish up your PhD. It's a fairly slow process, but eventually the article gets published. It meets all of the editorial standards and it's published in a journal, and you're famous, you're infinitely famous, you're now an author. Okay, so back to how to choose a great paper. How do you know what paper to choose? I just told you how I got really fed up with people in my lab for choosing poor quality papers. So how do you make sure you don't do that? Well, there's multiple ways to do it. One is you can use PubMed to search for papers in your area. So you use a keyword search. So I would look at telomeres or telomerase, or might look at telomerase and cancer, or telomeres and cancer, or telomeres and replication, and look at the papers that come up. You can also look at news and views articles or commentaries in top journals. I'm going to show you an example of that in a minute, where they highlight certain papers. If you look in like the Nature or Science journals, at the front of those journals, they'll have little sections that talk about papers that they think are really important. They'll have little commentaries or news and views that discuss these papers. And these are not necessarily papers that are published in that journal, but ones that they think are very important. And that's a good place to look. Top journals, impact factor, I don't like that so much, where there's this judgment system where some journals are of higher impact than others because it's felt that they publish papers that are more of higher impact. You can go that route. Though in general, I would agree that some journals tend to have better quality science than others because it's harder to get through the review process. You can also ask your colleague or mentor for suggestions. So when I have newish students in the lab, I don't expect them to come up and find a paper. I expect them to take a look and see what they can come up with, and then we'll take a look at these papers together and decide on one that's suitable. Before you finally decide on a paper, be sure and read it carefully, because I have, especially for class, sometimes I'll be in a hurry and I need to find a paper that's a bit outside my area, that's relevant to the class material that I've been teaching. And I find a paper that on the surface looks great. It looks just right. And I assign it, and I have come up with a bad shock when I hadn't read it carefully enough. And I discovered that, oh, good heavens, there's all these problems with it. Or even worse, it's full of all this bioinformatics, for example, that I don't understand. You know, did I really want to choose this paper? So be very careful before you tell other people what they have to read, or before you advertise what you're going to be presenting. Make sure it's something that you really can deal with. Okay, this is what I was talking about, getting ideas from perspective articles. So here's the New England Journal of Medicine. Look, here's perspective. And here's one, focus on research. Let's not put all our eggs in one basket. What that's actually referring to is this scientific paper on a drug, a clinical trial about treating patients with a particular type of mutation. And the basket is a play on words because it's a clinical trial called a basket trial. So we're going to focus on these two papers in the next little bit of this talk. But this is a perspective and talking about this research article here. And so that's a good way to find a paper that's of interest. Then the next thing to think about is, who is my audience? How well do they know the subject matter? In other words, how much background do I need? The other thing is, will they have read the paper? So in some of the journal clubs I've been to, only the presenter reads the paper, whereas the ones in my lab meetings, often all of us would have read the paper. In which case, if everybody's read the paper, everybody will have read the introduction or the background section. And so if your job then is to delve more deeply into background that might be in other papers, or sort of filling in the extras, as it were. Yeah, so if they haven't read the paper, you need to give detail. If yes, you give the expanded background, and you focus on the concepts, the questions that have been raised, and the take-home message. Okay, it's all about preparing thoroughly. So look things up. If you don't understand something, neither will your audience. So look up other papers, look up, you know, the references that are given in the back. That's a great thing to look up. If you don't understand those, go to Wikipedia first, Google methods, and then go back to the bona fide journal articles. Look things up. Another thing you can do just to add some interest is, here we're coming back to this paper, this clinical trial, where vemurafenib, I can never say these drug names, vemurafenib in multiple non-myelomas cancer with BRAF V600 mutations. And here's the list of authors. It is the senior author who sort of spearheaded the research, probably had the funding for it, designed the project, was called this guy called Jose Basilega. Well, you know, you can add a little extra human interest or commentary. You might introduce a key figure by introducing a picture or bio sketch. Look here, here's the guy. He's at Memorial Sloan Kettering. He's editor-in-chief. A physician-in-chief. This is what he studies. This is where he's from Spain. Just brings things alive a little bit more. Okay, then back to the hardcore science. You need to present the background clearly, and you need to pose the question clearly. So papers are going to be trying to answer a particular question. And you need to identify what that question is, and you need to point it out to the people you're presenting to. So let's talk about understanding the background first. Where do you go? We've talked a little bit about Wikipedia, reviews, commentary articles, and the paper's introduction section. So, for instance, the paper that we're looking at now, this vemurafenib in multiple non-melanoma cancers. Well, what's vemurafenib? What's BRAF V600? What does the mutation do? And why is this focus on non-melanoma cancers? You know, why? Why? What's important about this? So this is the little commentary that was in the prospectus section, and it's talking all about, it gives some nice background, and it gives a background on clinical trials, and then it's talking about the new clinical trial approach that's actually being used in this paper. Here's the introduction section from the paper itself. It talks about what BRAF does, and the mutations occur in approximately 50% of cutaneous melanomas. And it also talks about it being present in non-melanomas, that their mutations are also present in other cancers. It talks about vemurafenib as a drug, and it's used to treat melanomas with this mutation. So then the question is, what happens? Can it be used to treat the BRAF mutations in other cancers? And then this is talking about this experimental design, these basket trial experimental design. It's a new type of experimental design. So this is where the background is laid out. And so in the journal club, you need to explain the background. So for this paper, you'd be talking about how the different tumors were categorized, the normal markers like BRAF mutations or other mutations that the tumors commonly have, the types of therapy that's normally used, and a whole theory that we don't have time to go into now about the importance of tissue of origin. And that is that basically the concept is that to treat a cancer, how you treat it depends on if the cancer started as lung versus breast versus brain, for example, that you would treat it differently depending on where it started. And the whole concept for this paper is that that actually might not be the best thing. So as you go into the background, pictures are worth millions of words. So include pictures, include little cartoons. Here's a bit about BRAF, what BRAF does. BRAF is involved in a whole signaling pathway that's important in cell proliferation. You don't need to know what it does now, but if you're talking about it and giving the background, here's a nice picture. This drug, this cancer drug, here's the molecular structure. Do your audience need to know the molecular structure? No, but it kind of brings it home to them. When you show a picture, it's they kind of get their eyes around it. Oh yeah, this is the drug. It's a small molecule, right? So show pictures. Then a key part of the background is this, remember this last paragraph of the background or introduction section, how it lays out the paper's rationale and therefore what the experiments were about. And the rationale for this paper was that recent studies had raised the possibility that properties of tumors might be defined more by their molecular markers than the tissue of origin. Sorry, my phone's going off. And then, so this would be the first part of the last paragraph of the introduction, and then this would be the second part of that paragraph. To explore the idea, these investigators devised a new clinical trial approach. This is the basket trial approach to test the effect of this drug on tumors from different tissues that share this mutation. So this is really setting it out for the audience, what this whole paper is about. The question, will non-melanoma tumors bearing the BRAF mutation be responsive to the BRAF inhibitor, this vemurafenib drug? That's the sort of question that the whole paper is about. And then you would go into the background and talking about how the basket trials work. Here's a cartoon, we're not going through it, but you get the idea that you're showing pictures to explain this. It's all about preparing thoroughly and addressing the background, posing the question clearly. You don't have to present all of the figures, you present the important parts of the paper. So you have to judge what's important about the paper and present the most key figures, and you might just mention briefly the less important parts. So this means you have to actually read the paper very carefully, and you want to find out the details. You need to know the details, you know, what patients were excluded, are these standard criteria for a clinical trial? Is it okay to have no control groups? Here's how the methods, how the work was done. This is study design and treatment. This is the rationale for the work. This is more of a study design. So you need to really understand this so that you can decide what are the key points that you might want to make. And there'll be some bits that you get to, what? Statistical analysis? I have no clue. And you have to look that up. This was a whole thing about waterfall plots. What on earth are waterfall plots? You know? So it's up to you as the presenter to look this up, and so that you can explain it to other people. And it all takes time, but it's worth it, because then you can go into the presentation feeling really comfortable that you know how this work was done. So if you don't understand something, look it up. You go to papers, Wikipedia. Here, this is the statistical approaches and the waterfall plots. Waterfall plots, what on earth are those? Well, it's just a statistical approach and a way of analyzing the data. And so So I said you're not going to present everything. You're going to highlight the key pieces of data. So here's figure 3 with a key piece of data. Here's a table. Here's the key piece of data and the key conclusions. So what you want to do, it's a bit like I said earlier, read the paper, make notes, and then ask yourself, what's the big picture? Why did they do this study? What did they find? Why is it important? This is what you want to convey to your audience. And then to explain it, you're going to need to get into some of the details. How did they do the study? And what do the different figures and experiments show? And then you want to give your opinion. Did you agree with these conclusions? Did you see any problems? You want to present the conclusions of the paper very clearly so that people have a take-home message. And then you also want to get feedback from your audience. There's nothing worse than sitting there, everybody like bumps on logs, and nobody participating. So to stimulate discussion, you can ask your audience questions. So for example, here's one of the figures. You might ask, why do you think different mutant BRAF tumors might differ in the response to the drug? What ways might we use to predict which tumors would be responsive? As you'll have questions that will come up as you really study this paper. And so you go in, the question was, how do I give a great journal club? The answer is you prepare well. And then you're confident, you know your stuff, and just keep calm, and the big guys will back off because you know your answers. So I'm going to stop there. I have a couple of questions here. Yeah, does the Megan to everybody, with Christina answering partly. Does the order in which the authors are listed on a paper mean anything? And the answer is yes. It means a huge amount. The convention varies slightly from field to field, interestingly. But in my area, in molecular biology, usually the first author, as Christina said, is the person who did the most experiments, and really often is the person that will have done a lot of writing on the paper, a lot of experimental design. The senior author is usually the head of the lab. The person, they may not have done any experiments at all. They may not have had time to work in the lab, but they're the one who conceived the project. They wrote the grants, they got the funding, and usually they will do a lot of writing on the paper. They're the one that will send it out to the editors. They're the one, basically, they're the ones that stand behind making sure that all of the work was done right. And then the middle authors will have done different amounts of the work. You also have a corresponding author, and usually that is the head of the lab, the last author in the paper, the senior author as they're called, because it's their lab and they're taking responsibility for the paper. But sometimes when I've had more senior people, postdocs in my lab who were about to go and establish their own labs, and they have really played a very key role. I mean, they've really directed their own research. Then we may have co-corresponding authorship. And so that signals to people that they really, they had an equal responsibility in producing this paper, and people who have questions should contact either them or me about the paper. Any other questions about this? Think about. How many of you are going to have to give journal clubs you know of? Some of you are. Yep. Yep. It just takes a bit of practice. Yep. Well, you've got quite a number of you. I mean, it's just the way It's the way that science is communicated. One of the ways that science is communicated, and it's a great way to get a group of people together to really stimulate discussion in your field and to learn new material. Any other questions? In that case, I'd say give me a show of hands again who thinks they might be coming to our the next session where